I'm Deanne Erdman with Baylor College of Medicine. Milestones in Microbiology is the 7th activity in a unit entitled The Science of Microbes. And the activity, of course, is aligned with the national standards. In this activity, students are going to consider the relationship of historical events in the history of microbiology. Materials are pretty simple. You're going to have some cards that you've cut out from the masters provided in the template of the PDF, which is available with this presentation. Timeline, which the students will tape together. Cellophane tape. Scissors. Paper clips. And colored markers. And once again, we always recommend having some sort of nice dissemination method when you're working with students so that there's an easy, ordered way to pass out materials and for materials to be returned in condition for the next time you want to use this activity. Safety. In this particular activity, there's no unusual safety concerns, I guess, other than using scissors. But once again, you always want to bring that to students' attention to at least think about, are there any safety concerns when I'm doing an activity? To begin with this activity as a focus, you might want to ask students, how does one discovery or invention lead to another? And you may have to prod them a little bit to think of some examples, but you might give them, once you give them one example, I think they'll think of some other things, and many of them will be more modern examples, maybe related to the internet. But you might talk about books as a way, and print as a way of being able to communicate, but without the printing press, then that was not possible. Discovery of planets in our outermost solar system, not possible without telescopes. And related to this particular unit of microorganisms, you think about lenses and microscopes and being able to see microorganisms and be able to understand them a little bit better. So you may want to brainstorm a number of ideas from students and get them thinking about that. That's really one of the things that hampers new discoveries is the technology that go along with them. Now, each discovery reading summarizes an important scientific discovery related to microbiology. What students will do is to within a group, they will read, they'll take one card, they will read, and they'll highlight some important key points that they think will be used in determining when this discovery was made in relationship to others. Once they've highlighted some key points, and they say the first student is using an orange highlighter, then they pass it to a second student who maybe has the pink highlighter, and they will highlight anything that they see that's important. They may agree with the first student and just highlight over or underline what the first student did. And you want to continue this process until everybody in the group has had a chance to read every card. Then once they have all the cards and they see kind of what the common threads are that that different group members have highlighted, they'll be able to put it into some sort of ordered sequence so they can line that out. Next, they want to match that to the timeline that they've taped together, and that's where they may want to use the paper clips to clip on a particular event that they think had to be the first in this series of historical events related to microbiology. And they may say that this had to occur probably in the 1600s, and then so forth and so on along the timeline. Then each group gets a chance to, and you can make it a contest to make it fun, or you can just have each group take one item and say what place in history they thought it happened and why. And then as a class, you're going to have some differences and have the class discuss why they put something in a particular and defend that position and why someone else put it in a different place. So once they have taken, once they've got it all laid out, they'll be able to see why some discoveries took a while and until the technology caught up with the science that looked at that particular phase of microbiology. Once the students have finished laying out their timeline, then the next thing that you want to do is to have the class to decide what they think. And there's a couple of them that are really pretty close together that kind of could go either way based on the clues that are given in the reading. And so they want to see if they can iron out those differences. May even want to do a little bit additional research to see if they can figure out when something occurred in the timeline. And as they're talking about which particular point in history they think a particular event occurred, have them give some examples of what was needed before this could occur and maybe what a next step might be. Take time, once again, take time to examine this as a class, and then what you may have students do is to research some interesting information about some of these different discoveries to make it more personal. See if they can find out, almost like the media today, something that makes it a little more personable, maybe something that would make it newsworthy. They may want to calculate the time that it took between different events. Some of these discoveries happened very close together and some were much further apart. Why do you think that might be? At the conclusion of this activity, you may want to have students debate some of the merits of the Internet and the instant dissemination of information and proprietary rights to that information. And certainly there's a lot of academic freedom where we want to share things, but at the same time, it takes money to run laboratories and to build equipment. And so that has to be a consideration as well. So it might make for an interesting classroom debate. I think your students will enjoy this activity, especially if you let them get into it a little bit more. Thank you.